in discussion in the sense of like, here is a primer that then makes the sequence, right? So I think a lot of people make two kind of common mistakes are fine, like that's the reason to do a quiz like this. You can see how comfortable you are or not with PCR. But I think a lot of you actually have memorized already that picture of what PCR looks like. So it was really right where you have kind of the split and the two primers. So it was really interesting to see the relationship of that with the relationship of this one. So before getting into antigen, I just wanted to do a small thing on PCR and then primers. So the question asked you, or gave you 50 base pair sequences, and said amplify 40 with 10 base pair primers. I'm not going to write out the whole thing, but we can write out an example. So let's say, this is our DNA sequence, right? Well, a 5' in and a 3' in. So the things to think about in PCR is that for dideoxy sequencing, like when you're thinking about the sequencing, the primer was something you were feeding it, so it was not included in that sequencing count. But that's like a special case of PCR. This is asking for like a more general phenomenon. So in a more general case of PCR, primers are included in the length of the product. So if you have a comment on your quiz, you look at the it's like not big enough, because most of you put your primers at the very end, that would generate a 50 base pair product, because primers, and hopefully you'll see if I draw this out in a way that makes sense, you'll see that it gets incorporated into the PCR product. So that was the main thing. And then the second thing were primers pointing in the same direction, right? Like eventually, each primer has an individual, it interacts individually with the template strand, but then gives you a double-stranded product. You need them to point towards each other to get coverage of that whole length. So if you have a comment about like wrong polarity, or some of them I tried to find where your primer was in the sequence and drew arrows, it's because it's not pointing in the correct direction. Okay, so this is our PCR. How long is it? Let's see. 5. So it's 15 long. So let's make a PCR product that's 13 long. So let's not include those last two pairs. So let's make primers that are 5 base pairs. So how I approach a problem like this is I think about, again, primers have to point away from each other, or actually, towards each other, not in the same direction, and they have to be incorporated in the total length. So if I want a 13 base pair primer, 13 base pair product, 5 base pair primers, I would do this. 1, 2, 3, 4, 5, pointing this way. 1, 2, 3, 4, 5, pointing this way. So then my whole product will be from here to here, 13 base pairs. So this isn't the sequence, it's the sequence of one of them. You can choose whichever polarity you want, but this will look like the picture of PCR that you end up drawing, right? So one of these primers can be 5' PG, CCG, 3'. The other primer will be the reverse will be this complemented. And you'll see why. So the other primer is going to be G, C, E, A, G. So how does that make sense? So this is what a correct answer would have looked like. That you get the correct size and the polarity that they're pointing towards each other. So how does this make sense? It's because there's two strands, right? This is showing you a single-stranded molecule. The DNA is always two strands anti-parallel. So in reality, this primer, this TG, CCG, is going to amplify off of the strand of DNA that will have T, A, T, G, G, C, right? It's going to be this is the 5'. That would be 3' over here, right? It's the reverse complement, it's the anti-parallel strand of the one I gave you in the question. So that one, and this primer is going to prime this way, right? Or it's going to synthesize from 5 to 3'. The other primer, G, C, T, A, G, right, can go off of that original strand that I gave you. there's a T that is not priming, and there's a C, G, A, B, T. This is 3 prime, okay? So you still have 4 strands in that very first step, is primers binding on 2 strands and synthesis to create 4 total strands. Does that make sense? So you're not going to see primers like this again on the exam where you have to generate primers, but this should help you think about when you're drawing that picture of like the different cycles of PCR, this is what's happening, right? Even though I give you a single stranded thing, you can think about it's really double strand, they have to point in the opposite polarity, so you're amplifying across multiple pieces of DNA. So when you look at the key and you look at the comments, those are the most 2 common things. So a lot of people would put another primer going this way. So you were recognizing that they had to be on the opposite kind of parts of the DNA because the polarities came the same way. Right? A PCR like that would never meet in the middle. It would never have overlap. It would just kind of amplify non-consistent pieces of DNA. Any questions on that? This is, you never really need to do this unless you're designing a PCR experiment, but I think it's really important to help you think about how PCR is actually working kind of mechanism. Okay. So let's switch over to the cancer genetics for a bit. Every time I switch, it wants to. There we go. Okay, so cancer genetics. So, most people are affected by cancer one way or the other. Personally experience it, know someone who's experienced, so I'm not going to, it can kind of be really interesting, but I'm also going to try to be sensitive in how I talk about it. I think regardless of the personal health impacts, it's a really fascinating aspect of biology just because it gives so much insight into genetics, evolution, so many different things. So if you're personally affected by it, maybe it will help you think about it. If you're not, maybe you can just think about it in terms of like a synthesizing kind of topic in this class where you can kind of piece together all the different things you've been studying. So now this picture on the screen is really hard to see. It's little clusters of dots that are colored differently. Has anyone seen a figure like this? Or have a guess at what this might be? No, okay, that's fine. I don't think they had a picture like this in your book for cancer, but they did for other stuff. This is a plot of RNA-seq data. So we talked about RNA-seq data when we talked about genomics. We can get RNA out of cells, sequence it, turn it into cDNA, sequence it, and then what are you going to do with that? You want to kind of make patterns. You want to identify cells that are having different regulation, gene regulation compared to other cells. So this is like a 2-dimensional plot. You don't see the axes because people like to just draw like this. And basically each dot is a cell, and somewhere in here there's a big massive cancer. You can identify it through differences in gene regulation. For this whole kind of half the class, epigenetics development, in cancer, we've seen how important gene regulation and differences in gene regulation are to cell kind of morphology, right? You can think of cancer just being a special case of developmental and epigenetics, where the phenotype is something bad, right? This cancer is tumor like growth. So how does cancer kind of come about? So we're going to talk about that a little bit. I'm not a cancer biologist, so I only know kind of what I know, but we want to understand and kind of get on the same page about this so that we can then talk about genetics. So this is a figure from your book, and it's trying to highlight the role, or it's really, there's a complex set of genes that are important for cell growth and regulation, and the one at the center is kind of P53. Your book called P53 is this big green dot. So this, how do you read a figure like this? This is a figure of protein interaction. So each circle is a protein, and then each line or edge on the network graph is an interaction. So, down here, there's kind of proteins that interact in kind of little units. But then what do all of these little units have in common? They all interact with p53. It's kind of a master regulator. And so your book calls it the guardian of the genome. So these other kind of colors are the different functions, both metabolic growth properties that these proteins are involved in. So if you just kind of, even if you can't read it, if you just look at all the colors, you can think of how important p53 is to cell cycle and cell growth. So how do we go from kind of knowledge of protein interactions to cancer genetics? Oftentimes it's by how cancer is diagnosed, right? Someone comes in, they have, a condition, it's kind of unique to them, but oftentimes you can actually trace cancer in the larger sense through biology. So this is actual pedigrees from families with Lee-Fraumeni syndrome. So this is also from your book. So you guys are all experts at reading pedigrees by now. What do these pedigrees look like to you? Or what pattern do you see in these pedigrees? You guys are drawing cancer pedigrees for your Draw to Learn this week too. Yes, sorry.
Probably autosomal dominant in most of the cases.
Yeah. So these are different families, but what they're all showing is autosomal inheritance. XY squares are passing it on to circles, XX causing just as frequency as XY causing. So both sexes are passing equal sexes, so that gives us autosomal. And the dominant, because most affected individuals have an affected parent. That's not always the case. So what's going on here? There's these affected parents that are not having anything happening here. What are these diamonds? We've never really seen diamonds.
I was adopted.
That's the thing about cancer, right? Like cancer, we often have the phenotype, you have cancer, but what's the thing that geneticists care more about than the actual phenotype of cancer? Your risk of cancer, right? Most of the time when you're doing genetics, you're not finding a gene that causes cancer, you're finding a gene that's associated with an increased risk of cancer. So I forget what the diamonds mean actually, but that's why there's some kind of missing parts where not all of the presently even affected parents have it. Maybe they're just too young. Maybe they haven't reached the age where you start to see some of these cancers, right? Because there is kind of an age relationship to a lot of cancers. Not all the time. So this Lee-Fraumeni syndrome is actually due to p53 mutations in the germline. All right, that's the other really interesting thing about cancer, and the point I want to make with the slide is about what's being inherited, right? The cancer is not what's being inherited. Most cancers are in somatic cells, right? Cells that are not going to be contributed to the next generation. So it's not the cancer that's inherited, it's the risk factor. It's the risk increase of having cancer that's going to be passed on. So this pedigree is showing a mutation in p53 in germline, such that individuals have this mutation in p53, so they're passing on increased risk of cancer, because if we go back to this slide, p53 is the guardian of the genome, right? If you don't have p53, or functional p53, you have this increased risk of cancer. So does that kind of make sense? Kind of, we want to change how we're thinking about cancer, and that you're not inheriting cancer, you're inheriting the risk. Does that sort of make sense? A diamond, so someone helped me out on Twitter. A diamond is if the gender is not yet known. Yeah, I think those could be in utero then, I guess? But you know the gender pretty early within that. But anyways, that's the main take-home point. This pedigree is also not showing, it's showing Lee-Fermini syndrome, but these individuals, if you read in the book, have lots of different types of cancer. Breast cancer, sarcomas, neoplasms. It's not one type of cancer that's being inherited. Again, it's that risk, the risk factor. Some cancers do look inherited, right? But there again, it's due to risk factors, not cancer itself. So how are you kind of geneticists? You know, we have lots of definitions for cancer that we use in kind of our everyday life, but what would a geneticist call cancer? What would a biologist, a person studying cancer, decide on? So cancer is a group of disease characterized by cell proliferation. So that cell proliferation is often how cancer is diagnosed, because that's the tumor formation, right? But if it's a genetic disease, you know, we can think of cancer as a genetic disease. What's going to be true about this? What do we have to be able to take into account, especially in human genetics, if we want to think about genetics of cancer and risk? And you saw this last week in discussion. Right, we had a problem where we discussed the risk of, I think it was colon cancer in Utah or Shanghai. You had to think about an experiment, how you tease those apart. If you think about cancer, like you can think about any phenotype that's quantitative in the sense of like, it's not, yes, you have it, no, you don't. It's like, there's an 80% risk, a 50% risk. It's a quantitative trait. It's going to be influenced by both genetics and the environment. So you really have to think about the role of environmental factors in cancer. Those exogenous environments can influence cell cycles, influence DNA damage, and we'll see how mutations accumulate that are going to eventually lead to cancer. So this is just another example from your book. Your book has, you know, the whole chapter of these kind of different types of pictures showing different cell proliferation. So the main thing about cell proliferation, like, why is cancer problematic? Like, okay, cells are growing, sure, but it's how they're growing and what the consequences for the rest of the cells kind of in that little microenvironment. So here, the point of the figure is that the tumor cells, due to cell proliferation, crowd out the normal cells. Right. That's where we'll get to some of the other terms for like a benign tumor can grow. It can grow to a large size, but usually when people are talking about cancer, it's really a relative term based on the properties of how it's affecting other cells in the body. So I think here in this micrograph, the dark cells of the cancer here, oh yeah, they have them labeled kind of below on that other one too. Oh, this is liver on the top side. And then the second one is lung. So that's the main take-home point here. So we're going to think about genetics. We have to think about environmental impacts. It's not the cancer itself that's being inherited, but risk factors. And it's cancer when it starts impacting kind of normal cells in that local life. So this is again from your book. You know, how prevalent are these different types of cancers, right? Really, really prevalent. But again, some of these cancers do have unique risk factors, but some of them don't. Like there are generalities that we can pull out of cancer. That's one of the hard things about studying cancer, but also one of the interesting things, right? Each patient is kind of individual in how they oftentimes accumulate these mutations, but there are commonalities that we're seeing in terms of cell proliferation kind of hijacking that cell cycle. So when you see these statistics, it can seem kind of overwhelming from a genetic standpoint, It's pressing from a human health standpoint, but I wanna, as we go through the rest of this lecture, this isn't like definitive, these are the answers to solving cancer, but how can we make generalities and think about cancer differently? So here's some different definitions that your book uses to try to talk about the different stages of cancer, which I think are important, or like the different types of tumors that can be classified. So we have a benign tumor. So that's where the tumor is localized. We have malignant tumor. So the cells invade other tissue. And then metastasis. So this is where they induce secondary tumors. We've seen the word induction several times. And what does induction usually evoke? A change in gene expression. Genes that are not expressed being expressed. Right? So genes that would normally not be leading to cancer, something's happening, expressing, leading to overgrowth. Right? It's a developmental phenotype. That's the other main point I want to make about cancer. At the cellular level, it's a developmental phenotype. Any questions on this? So how are we going to start trying to tease apart the genetic aspects of cancer? Well, we can just look at kind of the techniques we've already used. We can look at patterns of inheritance. So pedigrees. You can look at the chromosomes of individuals. You can isolate cancer. You can do a chromosomal swap. We've seen the effects of mutations at chromosomal level, but duplications and versions deletion. So people have done that before. You can see chromosomal abnormalities. These are kind of correlative, right? The correlative patterns. How do we know that cancer is just not all environmental? Because we see these effects on patterns that we know have genetic components, right? Chromosome shape and structure, inheritance patterns through pedigree. But we want to move to kind of a mechanism. So these are the two mechanisms that are discussed in your book that we're going to go over a little bit. One was Knudson's multi-step model. The key point here is that oftentimes it's not one mutation cancer, right? There's sequential mutations that lead to cancer, which implies something. It implies that a certain type of kind of cell proliferation and cell tracking is happening in the development of cancer. And that's called clonal evolution. So maybe using the term evolution or selection is kind of a misnomer. It's not how I define evolution, but that's the phrase that your book likes to use, so I'm just putting it up here. It is, I wouldn't call it evolution because it's not a population, but it is evolution if you want to think about alleles changing, right? The mutation is increasing in frequency through clonal propagation, right? If a cancer cell, a lineage of cells, has increased cell growth, it's going to outcompete basically the rest of the cells surrounding it, increasing the frequency of that mutation. So here's the Knudsen multi-step model. So the main point here is that it's multiple mutations. It's not usually single mutations. And that really changes the way that we might think about cancer and the inheritance, right? To me, that makes us think about risk. If there's a protein in your cell, so we talked about P53 in discussion last week, we talked about helicase associated with Bloom syndrome and increased cancer risk. Right? If there's a mutation that can increase the likelihood of future mutations, you see a higher evidence of cancer. So in this model, they're just trying to show the different alternatives, right? If you, on the top is kind of somatic mutations that are happening sequentially, right? So in the top, the mutations happen in the same cell lineage. And then the bottom one, mutations are happening in independent lineages, but you have this predisposition. So you have like an initial mutation, and then secondary mutations are in independent lineages. They both have the probability of leading to cancer. If you have two mutations, it gives you cancer. But which one would you say is more probable?
Second
Yeah. So you could do a probability, right? The probability of a mutation 1 and the probability of mutation 2 occurring in the same lineage, if mutation rates are low, would be really, really low. But if you have this probability here in the bottom case of this initial mutation that just increases the rate of mutation or predisposes any mutation that would lead to cancer, you're already increasing the likelihood of cancer. Right? So this is like a hypothesis. This was a working model that was based on data presented at the time, showing that there were often different conditions that were kind of prerequisites to developing. I think this was eye cancer, right? The glioblastoma. That was based on this model. So if you're looking at this graph, the main take-home point is thinking about the probability of a mutation occurring that leads to cancer, because the data were telling scientists when Nelson developed this model that you need sequential mutations. It's not a single mutation, you need two mutations. I just also wanted to start introducing kind of the concept of cellular lineages, right? So you have one progenitor cell, and individual cells in the tissue are kind of undergoing their own mutational accumulation, right? These cells, once they kind of divide, they're not undergoing meiosis. We're in kind of somatic tissue, but you can think of them as having their own trajectory and accumulating mutation factors. So that's really important when you start thinking about clonal selection or clonal evolution. So in this model of clonal evolution, it's really just the extension of Medson's model. Medson says you need multiple mutations to get to cancer, and this model of clonal evolution just says, well, what's the impact of that? So multiple mutations allows cancer cells to aggressively grow and proliferate. So that's kind of what would be shown after these rounds of division. So you have the first mutation, the second mutation, you increase rapid cell division, and then now you have drug control changes in the malignant cell. So if you were to plot kind of over cell division, the number of cancer cells, right? Originally it's staying kind of flat, where it's at a very low frequency, it's kind of zero. It's kind of waiting for these mutations to accumulate, right? You're waiting. But once that happens, it can increase exponentially. So not only does it kind of look like evolution, right? We've seen graphs like this. Due to selection, we've seen allele frequency change if you have a fitness advantage. Again, I don't want to confuse evolution that's happening at a population level with something in a cell lineage level, but it's really, really analogous. And that's why the term evolution gets thrown in there, right? You're increasing the frequency of this allele. By increasing cell number. So for people who might have more of a background in ecology, it's the same thing of kind of like exponential growth in a population, right? It'll keep going until it reaches its carrying capacity. So there's a lot of analogies that you can use. But the main thing here is that if you think the tumor is spreading, it's due to this increased proliferation and increased allele frequency change, not due to changing over generation, but changing through the cellular lineage. Any questions on that? So we have all these underlying cellular processes that gets us at the genetic aspect, but what about the environmental aspect? What kind of data do we know or do we have about environmental impact? And the best data is about differences in incidence of cancer between populations. So again, we saw this when we thought about colon cancer in Yukon, Shanghai. This is just adding more numbers to it. And some of these differences are huge, right? So Switzerland versus Kuwait, the incidence of cancer, the difference in that incidence is astronomical. Okay. So now what I want to do is I want to transition to thinking about specific types of genes and mutations. So these are going to be broad classes of genes. So there are many genes that can fall into these categories, but they're going to give us very distinctive patterns of kind of genetic inheritance or genetic consequences for cancer. So the two main classes that we're going to think about are oncogenes and tumor suppressor genes, because they're kind of like at the end of the spectrum. One of them behaves one way, one of them behaves the other way, if you kind of just classify them in this binary way. So what's an oncogene? So this is kind of one of those cases in genetics where we define the function of the gene through a mutation. So I'm going to give you the phenotype of the mutated allele, which will kind of hint at what it's doing in the wild type scenario. So when mutated, they're dominant, and they stimulate genes that cause cancer. So where else have we seen dominant mutations that stimulate expression of genes? Can you say that again a little louder for me? Yeah, we saw it in bacteria systems. We had regulatory genes in bacteria systems that would give you constitutive expression, right? So we call those trans-dominant mutations. So we're not going to add the prefix trans on this, but I want that image to kind of do the same thing. A single mutation in this gene can affect expression at all other genes in the cell. So it's dominant, it's trans, right? This, whatever this gene is, has the ability to affect expression throughout the genome. So this is kind of the mutated case, so it gives us kind of ideas about what type of genes might become oncogenes. So we call these proto-oncogenes. So if we have a mutated gene that can change gene expression kind of at a whole genome-wide level, the wild-type gene must be responsible for a basic cellular function. So basic here doesn't try to imply like a simple function. Basic means like a necessary function, like something that has to happen in all cells all of the time, so that any kind of mutated, boom, right? Genomic increase in expression. Maybe a gene like p53. I don't know if we call p53 technically a proto-oncogene, but that thing, right? Something, I go all the way back to that slide really quick. p53 is connected to everything in your cell. If you have a mutation in p53, the whole genome will change. So that's the level that we're thinking of for these kind of dominant trans-like mutations. A single mutation affects everything else in the genome. So what about a tumor suppressor gene? So when they're mutated, they act recessive, and they inhibit, or they're inhibitory for genes that are inactive. So I said that kind of in a weird way. 2 mutations are needed at tumor suppressor genes, and now they are normally inhibiting genes that are inactive, and now they lose that function. So this definition is giving you wild type and mutant. They're normally inhibiting genes, and now they're inactive. So these, you can also think of them as trans, potentially, right? That in eukaryotic systems, trans cis isn't as important in bacterial systems, but the main thing I want you to think of here, and the contrast we're making, is that 2 mutations are required. Right? They're recessive. So this is a figure from your book that is trying to highlight the differences in kind of heterozygote or homozygote phenotype and function in cancer and in the wild type scenario. So on the left-hand side for the oncogene, you have a proto-oncogene. So it says it normally produces a factor that stimulates cell division. Now you have a mutant allele that's dominant, and it can increase cellular expression, right? So this is something that normally stimulates. And after, kind of like hyperstimulation. And that makes sense for a dominant locus, right? Like if we see dominant acting alleles, it has to be hyperstimulation. Most in eukaryotic. In prokaryotic, we saw super suppressors, but in a eukaryotic gene, we know that most genes kind of will always have a basal level depending on transcription factors. So if they're highly expressed, the dominant mutation is mostly going to increase expression a lot. These tumor suppressor genes are normally inhibit gene expression somewhere, and now you see the loss of inhibition. That's because this gene becomes inactive. So the same thing I said on the previous slide, but trying to give you that kind of mental image of oncogenes, you're heterozygous most of the time. The probability of that occurring must be different, right? And the probability of accumulating 2 alleles in the recessive case. So think about heterozygosity and think about probability when you're thinking about these 2 cases. So how do these mutations occur? They can occur in a lot of the ways we've seen mutations occur already, right? Insertions, deletions, SNPs. The only added thing that we're going to add back to cancer that we kind of ignored in some of the other sections are these translocations. So this is an example of a deletion. So how would you get kind of this heterozygous state? Or how would you get, well, let me just step back a second. What is this deletion doing for this tumor suppressor gene? So let's just read it, but we have a term that we can apply to it. So on the left-hand side, a person is heterozygous for a tumor suppressor gene. So in the heterozygous state, are they still have wild type function, or do they now have this kind of cancer-like risk?
Wild type.
They're kind of wild type. It's a tumor suppressor gene. You need to be, they're autosomal recessive. Or not always autosomal, but they're recessive actually. So on the left-hand side, you're a heterozygote. You still kind of have wild type function of that inhibition. Only one of your alleles is inactive. Now you get a chromosomal deletion. What's happened here? What's going to be the phenotype for like cancer risk on the right-hand side now? So I have a clicker question coming up next that's going to be for oncogenes. Well, if you tumor suppressor, then we'll do oncogenes. Any thoughts on this? Yeah, and why? Yeah, so what's their genotype? They're no longer heterozygous, what would we call them?
It's
Anyone else?
haploid.
I can't read your lips that well, no.
It's like haploid or something.
Yeah, and they're haploid, yeah, that's a really good idea. Haploid, we usually are thinking of like whole chromosomes. There's a really closely related term to haploid. They only have one copy.
Is it hemizygous?
Yeah, that's
they're
what.
hemizygous.
Yeah.
Right? So this was heterozygous. Big A, little a. This is hemizygous. They have like nothing and then a little a. We said that tumor suppressor genes are recessive. In this hemizygous state, we see something that we saw last week in discussion as well. We saw pseudodominant.
Oh, yeah.
Having, right, pseudodominance is when you have one copy of an allele, you express that phenotype. When you normally wouldn't, it's because you're lacking the other copy. So we're trying to explain, well, how likely is it, right? I told you for tumor suppressor gene, you need 2 mutations. That can happen by accumulating SNPs independently, but the probability of that is probably low. What's another way that we can kind of generate this?
TCR.
Through a deletion. So maybe you inherit the little a. You're not having a de novo mutation in your somatic cells. You inherit little a, but then all you need now is one mutation, potentially a chromosomal deletion, and you now have this tumor suppressor gene that is losing its inhibition, and cancer growth can be significant. Okay, so our first clicker question, we only have 2 of them, just to make sure we get everyone's experience here. Okay, so why are oncogenes usually dominant, whereas tumor suppressor genes are usually recessive? You can put like a couple word answer. So I have notes, right? I use presenter view, so I have notes. So I feel like someone just copied and pasted into one. You guys don't see all the answers, unfortunately, but I feel like someone was able to find my notes and copy and paste them, which is kind of fun. So this question, we're just trying to summarize kind of what we've talked about. Okay, so as you guys get your answers in, we can just, there's no like right or wrong answer for this one. I think there's answers that are kind of more consistent with the data than others, but there's a lot of good ideas here. So does anyone want to share an answer, or if they have another idea since they put this in? So we just had a slide on why tumor suppressor genes are recessive, so let's not focus on that one. Let's revisit what I said earlier about oncogenes being dominant. So why are they usually dominant? Okay, so this is what we said for tumor suppressor genes. And then draw a model that's, I'm not trying to make it look like a bacterial system, but I will just because of how I'm drawing it. Right, there's this gene A that's usually inactive. You have this tumor suppressor gene. Your genome has 2 copies. Right, proteins from this are inactivating it. If you have a mutation in one, you kill this one, you still have this other protein that can inhibit, right? So that's why tumor suppressors are recessive. So why on the flip side then are oncogenes dominant? So I think there were a lot of ideas about this in the topic. Because they're kind of like activators. They're increasing expression. So if you have another gene here, and you have your oncogene, normally both of these proteins are gonna increase over the basal level of transcription. And now you get a new allele, so it just increases more, right? Since you're already at kind of transcription is high, any increases can have the potential for like hyperstimulate. So that's why they're dominant, because they're playing that stimulatory activating role, whereas tumor suppressors are playing an inhibitory role. So those are the main take-home points I want you to think about for oncogene tumor suppressors. It's kind of like a compare and contrast. What's the kind of inheritance mode we would put on them? Dominant for oncogenes, recessive for tumor suppressor, and why? Because one of them is normally inhibiting, and then there are mutations that inactivate that. The other one's normally stimulating, and mutations can hyperstimulate that. So there's questions about these on your study guide. The type of questions you might see on an exam are, and does anyone know the most common kind of new way that people are trying to treat cancer? Yeah.
Is it hormone therapy?
Really close, yeah. You cannot not just give people hormones, but you can do something else. You can give them a new gene therapy. So on an exam, would gene therapy work equally well in both of these scenarios? The answer is no.
No.
Why not? Think about that. Gene therapy
Because-
will not work for both types of cancer, but on the oncogenes and tumor suppressors. So think about why that's the case. Okay. So we talked about cancer. It's really a phenotype of cell proliferation. We saw there's certain classes of genes, and there's not just one oncogene or one tumor suppressor gene. There's many genes that we can classify into these two camps based on whether they're normally activating or inhibiting stimulation. But at the kind of end of it, the mechanism always has to relate back to the cell cycle. So what type of genes are kind of there in the cell cycle? So we have genes that can kind of control the whole cell cycle. You don't have to memorize this. This is just, if you think about cancer, you might hear some of these terms thrown out. I'm just trying to give you kind of some context for where we're going in the cell cycle. So in the cell cycle, you often get cyclin-dependent kinases. CDKs, and then cyclins themselves. So kinases, what? They add phosphate groups, okay? You're changing the structure of protein by adding a group to it, a phosphate group. We have a G1 to S transition. A protein that's really common there is, so it's named for a very specific type of cancer, but it can play a role in many different types of cancers. Right? We often, again, we name things based on the mutant phenotype, because then that helps us understand where in the cell cycle it's happening. G1 to S is really important. S is the synthesis stage. That's where a lot of the mutations are going to occur during DNA replication. And then you have G2 to M transition. So there you have mitosis promoting factor. We have some other checkpoints as well, spindle assembly. Any mutations in any of these genes can lead to cancer because of how they're affecting the cell cycle. So this is just one example from your book. So this is, here we're looking at RB proteins, and we're thinking about the transition from G1 to S checkpoint. So the main take home here is that the protein controls the progression by binding to a transcription factor. Right? So RB protein binds to E2S and keeps it inactive. So I don't know what your book, if it puts the binary label on RBP, but what would we think? If we were to classify RB based on this diagram as an oncogene, a proto-oncogene, or a tumor suppressor gene, what are we going to say?
Oncogene.
So again, I don't think your book does this because the next step is that you have those cyclin kinases that are going to inactivate RBP and allow ETF to go to the promoter. But that's going to happen at a very specific part of the cell cycle. So let's say there's a mutation in RB protein that makes it inactive. Right? What's the consequence? E2F doesn't have to wait for these cyclin-dependent kinases. It can just immediately start going to the promoter and synthesize it. Will that happen if there's a single mutation or two mutations in RB protein? Two. So if RB protein would require two mutations to see kind of this bypass of this checkpoint, what kind of protein would we classify that as? Yeah. So again, I don't think your book does this explicitly, so don't memorize that RB protein is a tumor suppressor. I just want to kind of go through that mental exercise. So if you had a mutation, two mutations, right, an RB protein, then the transcription factor E2F could go directly to promoters. Therefore, RB is kind of like a tumor suppressor. And are people seeing the logic behind that? So we're trying to place now this regulatory mechanism into the context where cancer is going to happen by kind of overcoming the cell cycle. There's a checkpoint. This phosphorylation of RB protein only happens when the G, normally in a normal cell, will only happen in the G1 to S transition. That's when this process is occurring. Cancer doesn't want to wait for this process, right? It wants to get to DNA, make new copies of DNA to continue cell growth independent of cell cycle checkpoints. How could it do that, right? It's like, how could it do it? It's not doing anything actively. What mutations could occur that would have that as a consequence? Mutations in RB protein. You need two of them. If you only have a mutation in one, the other allele would still be producing RB protein. If you have mutations in both alleles, no RBP protein, now the gene that's normally inactive, the E2F transcription factor, can do whatever it wants, whenever it wants. Does that make sense, that logic? So here's just another example. So this is the example, we're at a different checkpoint stage now. We're at the G2M checkpoint and cyclin B. So this is a really complicated graph in your book. But again, I just want you to think about what's happening here. So it says that cyclin B accumulates throughout interphase. And then near the end of that checkpoint, the mitosis promoting factor, so MTF here is mitosis promoting factor, reaches a critical level, and then it kind of pushes the cell through that checkpoint through mitosis. So there's a lot of stuff going on. This bottom one is kind of the level of cyclin B, right? And it's correlated with the level of MTF. And then the top part is where they're occurring kind of in that cycle. So if you have a mutation, so cyclin B is kind of this protein. If you have a mutation in cyclin B, what's going to happen? Well, you have a mutation in cyclin B that increases the rate that it's produced at. So instead of this purple line, we're going to have, I don't know what color you'll be able to see. Right? If you can increase cyclin B much more quickly, you're going to end up like that. What kind of mutation does that look like? Potentially.
Insertion.
So I don't really have have an answer. I don't think there are mutations in cyclin B that do that, but to me, maybe that looks like an oncogene, right? The cyclin B has to reach a critical threshold to stimulate something, MTF is working with it to push through mitosis. If you're doing that more quickly, you can go through the cell cycle more quickly. You can also just mess everything up, right? As your peaks in different stages of the cell cycle from cyclin B now. Right? So if you have a mutation in cyclin B, maybe can push things through the cell cycle more quickly. So that's kind of what we're thinking about when we think about cancer. It's all centered on this context. It's like, right, organisms have life history, right? They undergo meiosis, zygote formation, right? The life history of cancer is the cell cycle. So think of that as like a life history. Right? So where are mutations going to occur that are going to change the life history of cancer? So those are changes that are associated with like mutations in single genes. For the last chunk of the class, we saw mutations that can happen not at the DNA level in terms of like changing regulatory regions or changing coding regions, but can happen at DNA that changes their epigenetic marks, right? Methylation marks, histone acetylation, some of those chromatin marks. So cancer can also be affected by these processes, right? Cancer is all about gene regulation at the end of the day. Epigenetics is all about gene regulation. So what kind of epigenetic changes are we going to see often with cancer? So the things we've already talked about epigenetics. DNA methylation, chromatin structure. You can see hypermethylation. So what would that mean? So methylation, DNA methylation, what's the link between DNA methylation and expression? Or what's the correlation? Sorry, I just can't hear you that well. Yeah. Methylation inhibits transcription. So if we put methylation here, and transcription here, negative relationship. So hypermethylation, more methylation than there would normally be in a region of the genome is going to mean less expression. Maybe this has a phenotype really similar to a tumor suppressor mutation, right? If a tumor suppressor mutation is inactivating a protein, that's what hypermethylation is doing too, right? It's basically inactivating the region of that genome through reducing transcription. Then we can get hypomethylation. Less methylation than we would normally have. So that would lead to the stimulation of gene expression. That sounds really similar to me like oncogenes, where we have a mutation that stimulates gene expression. Right? So the phenotype might be very similar, and that's what makes cancer really complex. The phenotype is going to be the cell cycle dependent kind of deviation. Maybe it's happening through mutations that affect proteins themselves. Maybe it's happening through mutations that affect methylation. And it'll give you very similar patterns, either lower expression or inactivation of the tumor suppression gene, or increased activation of an oncogene. They both will look very, very similar. So I kind of gave away the answer, but just if your top hat didn't work for the first question, just make sure to get your answer submitted for this question. So hypermethylation is thought to contribute to cancer by doing what? So let's start ruling some of these out. We never talked about telomerase, so let's rule that one out. Expression is all about transcription from DNA to RNA, so we can rule A out. We're not at the level of DNA replication. That's true. In the cell cycle, DNA replication is super important. But at the level of transcription, that's not the molecules we care about. We care about the molecules transition from DNA to RNA. Then we have inhibiting the expression of a tumor suppressor or stimulating the translation of oncogenes. So we just today talked about tumor suppressor and oncogenes, so if you're still kind of thinking about those differences, that's fine. You can answer this question based just on the first word, inhibit or stimulate. If methylation inhibits transcription, and you're putting more methylation out there, you're going to inhibit the expression of something. So the best answer here is B. Any questions on that? So the last type of mutation that we talked about, we've seen when we have that question about hemizygosity or chromosomal deletions or mutations at the chromosomal level. And so we have deletion. We saw how that can make something hemizygous, pseudodominant. We have inversions and translocation. And then we have aneuploidy. So there probably are a lot of nuance to the different effects that these mutations will have, but there's one commonality between. So inversions, remember, are flipping the orientation. So if we have A, B, C, D, the inversion would be A, C, B, D. Translocation is two different chromosomes that then swap genes. And aneuploidy is a whole chromosome being duplicated. So now you have kind of like two chromosomes when you normally have one, aneuploidy. So it's not like a duplication on a small scale, it's the duplication of a whole chromosome. So if the first one we think will most likely be associated with making alleles hemizygous, and then pseudodominance of tumor suppressor, what might the actions of these other types of mutations be? Any ideas? My idea, and there's probably, again, there's not one right idea and answer, there's just ideas that are maybe consistent with data. My idea is that all three of these are changing the gene expression. And why? Because they're probably changing methylation and chromatin landscape. What do I mean by that? If you're a gene, let's just say gene B is right by the centromere. We know that centromeres often have heterochromatin, really tightly compacted chromatin that's inaccessible to transcriptional machinery. So first, gene B is not transcribed tightly. Now it undergoes an inversion. It's removed from its location by the centromere out to a normal part of the genome, less chromatin, more expression, right? That's the same logic can happen for translocations. I'm on chromosome 2. This chromosome is really highly methylated. Now I'm on chromosome 7 with no methylation. I increase my expression. Same with aneuploidy. Maybe the cell only has so much proteins to regulate one chromosome. So now that you're duplicating the number of chromosomes, you're seeing differences in translation or transcription because the cellular machinery just can't handle that. So for me, the first one's all about kind of the genotype, hemizygous. The next three are all about gene expression changes because they're altering kind of the fundamental properties of a chromosome that regulates expression. So this is just an example of a known kind of cancer gene and these translocations. So a reciprocal, so these are reciprocal, kind of they're exchanging pieces equally. So a reciprocal translocation in chromosome 9, chromosome 22 causes chronic a type of leukemia. I'm not going to try to pronounce that. Hold on a minute. Right? It's altered the chromatin DNA methylation of these genes causing leukemia. So here's another picture of a chromosome spot from cancer. All of those are, not only is there a lot of aneuploidy going on, but there's a lot of translocation. So they found probes. We talked about probes really briefly, right? You can synthesize DNA that will complement other DNA, and you attach the fluorescent molecule to it. You can make probes that are specific to every chromosome in the human genome. So when you see chromosomes that light up multiple colors, it's because they have different chromosomes kind of all squished together. So I think this one from your book is a colon cancer. So there's a lot of different chromosomal abnormalities happening in the colon cancer. Okay, so we're going to wrap up really soon. Viruses. How can viruses, right? HPV viruses increase what? Increased risk of cervical cancer? How is a virus causing increased risk of cancer? Mostly it's a retrovirus. The retro is like viruses that require inserting into a genome of the host before being replicated. That insertion can cause mutations, right? Retroviruses, they insert viral DNA into the genome. And that in turn can cause mutations. Interestingly, this is mostly for oncogenes. It's not that you're creating a mutation that inactivates a gene. You're actually, viruses want to replicate really quickly. They have kind of promoters that increase replication or increase transcription. So if you insert viral DNA near an oncogene, right, you've now just made a proto-oncogene, you've now made an oncogene. So the example that's often used is HPV, human papillomavirus, HPV, and cervical cancer. So here's all of the different types of, not all of them, it's not like a master list, but some of the different types of cancers and what virus is associated with them. So this is just a graphical representation. So you can either mutate and arrange proto-oncogenes, or I think most often we insert a strong promoter near proto-oncogene. Proto-oncogene is the wild type allele, and then you convert it to an oncogene by increasing its function. So if you want to know what you need to know about cancer for the exam, please look at the study guide. Please come tomorrow for discussion. We'll do TopHat again, but I have no more questions for you to work through. It's all a review session. So if you do want to come, that's great, but just come prepared with questions. So that will facilitate the conversation about what you need to know about cancer. And that's posted under the discussion. All right, so thank you guys for being a great class. I really had fun this semester.